Hello and welcome back to Intro to Cognitive Science. I should pronounce that better. Cognitive Science. So, we've been a few places now, right? We have talked in depth about different information processing models, different ways that we've seen an information processing system could come to potentially live in the physical world, right? We've come at it from many angles, and today we're going to start an exploration of how all of this begins to focus on the brain itself, the neurology, the neuroscience, the dawn of the study of the actual physical brain, and how it brings an information processing theory to the table. Not a single one, just like what it contributes to the idea of information processing. So, until now, we've had all these different ideas for how information can get processed. You know, last time we talked about Mars' visual breakdown of how could I convert light in such a pattern into some sort of abstract internal 3D representation of light, or of the objects that light hits, rather. And a lot of times we end up with this question, and especially in Mars' case, well, okay, but how does the brain do it exactly, right? And this has essentially been in throughout the history up until this point in the timeline. It just kind of punted forward. The technology's just not there yet. We don't know for sure what is going on inside the brain, but that technology started to emerge. Maybe a little sooner than you'd think. So this is more of an aside, but check this interesting thing out. So, 19th century neuroscience. This is a machine that takes a shunt into somebody's skull, and what it detects is the change in pressure inside the skull in response to things that are happening in the world. That seems a little goofball, but when you think about it, there's got to be blood flowing to this organ to make it function. And the question was, is that, related to processing power? And we see it kind of was. Resting quietly in the top one, we just kind of the pressure inside the brain, inside the skull rather, is kind of constant. It's resting state. But then a clock strikes 12 in the bell in the room, and the pressure goes up. Oh. Doing some math, we see the pressure moving around a little bit. What is 8 times 12? That kind of stuff. There's no evidence that this measure was capturing actual raw brain activity. I mean, it could just be capturing, I'm doing something. I'm moving. I'm doing anything at all. But the idea is that this is how we start to puncture the edges of, hey, what's going on in there, right? And this is coarse. This is a very coarse measure. You're not gonna see this in any modern tools, but it kind of underlies the big questions of the future of, hey, when is this thing in use? When is a specific part of this thing in use? So, let's talk about this, the functional approach to cognitive science. Cognitive science has typically focused on what are the actual functions of the mind and the brain, more so than the physical details of those things, right? Just like in Mars stuff, how do I turn a light pattern into a 3D concept? How do I, what is the function of selective attention? When does it fail? When does it succeed? What's surprising about that? That's a specific function that the brain has. Language is a very complex and robust, but function, right? A function of the brain. So these computational algorithmic questions, and less on the implementation level. And because in theory, I think we've touched on this a couple of times, but it's always good to retread this one. All the functions of a given mind could be multiply realizable. So multiple realizability is the idea that the system can be built in many different ways, right? They might even be different materials. They might be different shapes and overall structures. But ultimately, when I say the word car, you get that most of the things you see on the road are car, right? But they're not a specific car. There's huge variety in the models and makes. Sometimes they have larger engines and smaller engines. Sometimes they have larger frames. Sometimes they're more sturdy. Sometimes they're tiny little smart cars zipping around. Sometimes they have room for 10 people. Sometimes they have room for one. They're all a little different, but they all satisfy the primary function of car, which is go to place, take me there, right? So this is the idea that even if we're just talking about a single thing like drive me to the place, but even a more complex thing like the way a mind perceives itself, the question is often about how the thing works, the function of the thing, more so than the features of the physical instantiation of that thing. But this, if you just rest on that, you miss some very important things. So let's look at this. I can run addition on all kinds of stuff. How might you add two numbers together? 27 plus 58, right? Most of us are trained through like the middle school, high school era to we can run that kind of stuff in our heads maybe. But the simplest way probably is if you got scratch on you, pen and paper. Or if you're got your phone open already, maybe the calculator function, right? On your phone. Right? But these are different things. These are different tools. As I said, we can do it in your head, right? And if it's small enough, you could do it on your hands. Addition is a function, but the way that it works varies wildly between the mediums that are chosen. It takes way longer to do it in pen and paper, probably even longer to do it in your head because you're trying to kind of replicate pen and paper, right? You're trying to solve this math in various different ways. That is important because the way I do things on my hands is going to be quite different than the way I do it on a calculator, and guaranteed, the calculator is not going to make the same kinds of mistakes that my stupid fingers will. Or my stupid brain, if I'm trying to do it all mentally, right? It'll still make mistakes if I hit the wrong button, but it does it differently. Can also think about another angle on this. Consider the heart. First of a shrew, right? Tiny little thing. Moves around a lot. Has to move a small amount of blood around a small amount of body. Has very different concerns in life. Needs a lot of burst energy to survive escaping a cat, for example. Compared to a human. The human heart is quite a lot larger than the shrew's. But it's still doing the same thing, right? It is able to move up and down in terms of its heart rate. What about the whale? A whale's heart, if you didn't know this, it's the size of a VW Beetle car. That beats 6 times a minute. It is enormous. And that animal lives a very different life than a shrew. It has different constraints on the way that it has to work. And yet, it achieves the same function. So, this brings us to the issues that happen if we just assume everything is multiply realizable, and that's just fine. Because just because a function works on multiple kinds of hardware doesn't mean that the specifics of its implementation are going to be irrelevant, right? A calculator is better at math than my fingers. There's very different capabilities and expectations out of these different things. If I'm telling you, hey, multiply 100 digits times, or 10 digits times 10 digits, Doing that on your fingers, that's not great. Doing it on a calculator, yeah, that's fine. I can do that. And moreover, just thinking about this, if you make a car out of wood, that's probably not going to be a very good car, right? Because we know things about the way that wood works. It doesn't like combustion, that's for sure. So we're not going to be using the same kind of engine. And if we're not using the same kind of engine, that means we're doing some wildly different things to achieve the same function. So, and we'll show an example of a wooden car, but the idea is, for a long time, it wasn't possible to examine those specifics that we know when we talk about a wooden car are very meaningful differences. But for a long time, we couldn't look at those differences. We couldn't figure out what is the structure of the material of the brain. And here's the wooden car, as promised. But now we can. We can look at using different technologies, we can look at the activities and structures of the brain. From all the way from single neuron recordings, so we can tap into a single neuron and see how it activates, right? When does it get a signal? When does it not? When does it fire? When does it not, right? But all the way up to broader things like PET or fMRI scans. We can scan the entire brain to say, hey, what is happening at every given moment in this brain? And so the emergence of these technologies signals a shift, and perhaps not away, but an evolution on, hey, so the classic AI style approaches of cognitive science as, hey, what are the functions of a mind and how might that all work? Into a little bit more kind of bound to the real physical reality of the brain, saying, hey, how do we bridge the gap between what the brain is actually showing us that it does and the way that we believe minds must function? And this brings us into the realm of connectionist models, which we'll talk about at length as we go forward. So, okay, let's talk about fMRI first. What does it do? How does it work? fMRI, functional magnetic resonance imaging. It uses a magnetic field, a very powerful one. Normally around this point, I have everybody ask, raise your hands, have you ever been in an MRI? They're wild. They are incredibly powerful magnets. They make you take all the metal out of your clothes and your body if you have piercings, because the magnet will rip them from you. You don't want that to happen. That's how powerful this field needs to be in order to detect the iron in your blood. Now, it's whether or not the iron is bound to oxygen. So, oxygenated blood versus non-oxygenated blood will help to detect these things. But the idea is that, hey, in your brain, where there's blood flow, there's likely neural activity. So this scan at the bottom it's different slices. So the way that fMRI works is it will scan one sort of slice of your head at a time, and it passes over you. It takes about 1 second to get a full scan of your head. And again, it's doing this all through measuring disturbances in a magnetic field. You don't have to know how all that works. That's just trust that is how it works. It is a fairly mechanical process. That's kind of the cool thing about all this. We can see the brain just by saying, hey, I have a big old magnet. How does it change when a person's in it, right? So the idea here is that this is kind of moving. I believe if you look at the bottom right, that's sort of the top of the head. You can see the eyeballs there on the front. And then the one to the left of it is a little bit lower, a little bit lower, a little bit lower, until we get, okay, the eyeballs are no longer visible. We're in some other chunk of the brain. And keep going lower and lower and lower until you're in the upper left, where we're no longer really in as much brain territory. We're sort of at the base, probably seeing a little bit of mouth, maybe brainstem. So, and then it goes back up, right? It keeps passing back and forth over the head to get these scans. So this is a cool piece of technology because what it's showing is, hey, there's blood is changing within the brain, and I can see that blood. So this allows you to investigate both when and where, I call it right here cognition, neural activity, we could say it at the baseline, but thus cognition is occurring within the physical structure of the brain. This is awesome because this is both the overall, hey, how much brain pressure is going on when you're asking me to solve math. At the aggregate level, sure, we can say, hey, how active is the whole brain? But it's also able to say, oh, wait, when I have you do this task, there's only this region seems to be lighting up effectively. And when I have you do this other kind of task, this region lights up more. This allows us the idea of localization of function. We can map in the brain where these different mysterious functions might actually live. Or the modern take, and it's best to inform you now, the modern take is more, it's not about where does one function live specifically, it's more about what regions, what networks of regions are recruited for a given function. Because ultimately, every function your brain does is very complex and composite. I think I've mentioned before, every little thing you do is full of complexity, right? You are having to organize a lot of things just to create one sentence, just to remember one detail of your dinner. There's a lot at play. So it's not necessarily about finding out, oh, this little brain region, that's where grandma lives. This is more about this little brain region is one computational piece of a bigger network of systems. If Sherdlu is anything to be believed about the way these things could work, remember, then to process language, we're going to recruit a lot of different chunks of our brain. They're going to be specialized to different things, and they're all going to come in and say, ooh, we can help with this task. Those same chunks of your brain are probably not going to be active when you're doing something like learning to juggle. Maybe some of them, but not all of them. So anyway, that's a bit of a tangent. The idea, though, is fMRI gives us this both where and when. That's super cool. But there's an issue with fMRI, and that is it's pretty dang slow. I like that it's 1 second at a time for each slice, but 1 second is an eternity in neuron land. So fMRI is great. It gives us this good where, very, very spatially situated. We can see all the brain and how it's reacting. But the when is kind of slow, right? 1 second is a lot of time for things to be happening in the brain. So we can use single cell recordings to shore that up. And we can say, okay, we can actually measure in real time the activity of a single neuron. And that's cool. I mean, so this involves having electrodes directly on the scalp to capture this. Build. Huh. Anyway, so it animates. Huh. Yeah. So we'll talk a little bit more about neurons. We're not going to get in depth in this course. We'll look forward to 266 if you want to really dive in. Well, dive a little deeper in to how neurons do their thing. So yeah, this is a tool. This is a tool in the kit, right? So single cell recording is nice because we can combine it and say, hey, we've got an idea of where things seem to be happening with fMRI at this really broad, high level. I can measure the brain and see, oh, these areas are more active when I'm having somebody do this kind of task. Then we can go in a little more deeply and say we can do some single cell recordings, right? So yeah, this is great because it lets us capture the processing in real time, right? As it is happening, saying, this part is lighting up in this exact pattern, not just a sort of a 1 second blur of, oh, well, over the last second, there was this much activity across everything. This is saying, I know where I'm looking. I'm looking at a very specific spot, and I'm trying to see exactly when everything happens. For instance, looking at how the visual system processes things. So let's take a look at the brain at large, do some of this favored mapping we've been talking about. fMRI, brain imaging techniques. The study of the brain goes back as long as we've had scalpels, of course. The physical look at what's inside the brain. But we've uncovered more as we've been able to actually measure the neural activity in the brain, right? So, in broad strokes, the forebrain is all about thinking, perceiving, evaluating, sort of these slower, more deliberate things, right? Whereas the midbrain might be more about programming motor activity, processing audio and visual responses, these kinds of things. And then the hindbrain is like that lizard brain type stuff, right? The balance, movement, breathing, heart rate, metabolism, that kind of stuff. So we see a little bit of a glimpse through time. The hindbrain would be the very first brain that would be required to survive as the complex organism that we are. The midbrain allows for a little bit more skill and expertise, and the forebrain coming later allows for kind of complex abstract thought, the kind of things we've been talking about. This is, again, just a glimpse, just a broad glimpse of the way these different regions or different levels of the brain work. We can also talk about, of the forebrain, we can talk more about the lobes on the highest level, right? So up in the very complex meat, right, the more deliberate, higher level function stuff, even that is kind of roughly divided into regions. In the very front is sort of the cognitive, the frontal lobe, much more, your deliberate thinking, your processing of symbols and complex relationships and logic and all that kind of stuff. And again, don't get too attached to all this. That this is not about, these things are already degrading. As I mentioned before, it's much more these days about understanding the way different networks of activity function together than it is about partitioning brains into special regions. But these things generally hold. So anyway, frontal is a little bit more cognitive in its nature. The parietal lobe has a lot to do with spatial reasoning, so understanding how to move through our environment. Maybe this is where Mars 3D abstraction would go to help us figure out, this is how this 3D object would fit into X place, or this is how I would use it physically, right? So parietal lobe is all about spatial stuff. Temporal lobe is all about processing audio. Not all about processing audio. There's also some memory going on there. But audio, right? It's the ability to process something stretched out in time. So processing the words I'm saying to you right now that you are hearing are getting processed in your temporal lobe. And the thing you're looking at right now is getting processed in your occipital lobe, the vision lobe, the area of the brain responsible for catching all of those light patterns and starting to do interesting stuff with them. Kind of that, the zero crossings, those kinds of things. Although those actually happen at an even lower level, like almost before they even hit the brain. But point is, this is kind of a general overview of these brain regions. And I like to do this often, just to get yucky. I mean, this is what's in your head. This is an actual slice of a brain. You don't have to memorize these parts. Just, this is a brain. This is what we're working with. This is the thing. This is the meat. This is the meat that thinks, right? This is how you do your thing. This is how you're doing your thing right now. It's a little bit, makes you shiver some, but it's kind of fascinating, right? So, this is great. We've talked about kind of how the emerging technology helps us start to understand, what is going on in the brain? And this is great. It leads to new investigations, and it's great to be able to sort of map isolated activity, like in the fMRI. But there's the bigger question of, how does information flow through the brain? How does it move through the brain? Well, let's roll back and think about a system we've already looked at this for a little bit, Mars' take on the visual system, right? Mar started with this top-down approach, where what's the flow, right? We take raw light patterns and we process them into primitives. That's one region, one processing element that must achieve that. Those primitives then get processed into surfaces. Another set of elements would have to achieve that. And then finally, from those surfaces, we get this sort of more abstract 3D concepts, right? So that is a flow of information that does some useful stuff by turning light into actual understandings and representations. Ungerleider and Mishkin come along in '82 and start fleshing this out further. And they have this two visual systems hypothesis. And it's more of a bottom-up approach, right? So Mars is saying, this must be how we are able to do this, because this is at the highest level. The logic of this shows this must be possible. I'm not sure how the brain does it, but this is something that the brain must be doing. Whereas Ungerleider and Mishkin come in and say, well, we're going to look at what the brain is doing and figure out how that might fit into this other higher level system. You see the multiple approaches, right? Sort of this AI and cognition guy compared to these neuroscientists that are connecting the dots on this very complex thing, the brain. Okay, two visual systems. The visual information that we get in the brain, into the brain, the idea of this is a big idea. This is not just one single study. It's the whole idea that there's not a single pipeline. This is the theory, right? There's not one pipeline through the brain that information comes in, do, actions come out of me. There are multiple routes. There are multiple ways that the information is moving, and it does different things at different spaces, right? The division of labor is the key idea here. So, two streams. The ventral stream, and ventral is sort of like the front. This is where, if you think about it like this, dorsal fin is on the back of the dolphin, right? Dorsal is on the top. The ventral is where you vent, your face, your mouth, your nose, etc. So ventral stream is always going to be towards the front of the body, and the dorsal is going to be towards the back. That's just a rule of thumb for when you're talking about these things, because it has to be body-centric the way we talk about them. So the ventral stream is the forward stream. So the visual information moves into the temporal lobe. And this idea is the same information is going from the occipital lobe to the temporal lobe, and it will be used for identification, but it's also that same information is moving up to the parietal lobe for spatial reasoning. So, as we've discussed before, this is sort of tapping into the same things that Mars' model was coming at it from. This is about medical patients, right? Patients who had damage to each of these different areas struggled with specific tasks, right? Temporal lobe damage meant they had difficulty recognizing the objects. They couldn't put names to them. Something about the way that their information processing, their visual information that they had processed, it hits the temporal lobe, and then it fails to access memory, the label for this thing. Whereas in the parietal lobe, if that is damaged, when the information arrives there, the temporal lobe is fine. It's doing its job. It's labeled the thing as the hammer. But now they're confused about how to use it because something in the parietal lobe is not functioning properly. It's not helping them process how is this thing used, right? The spatial reasoning stuff is all haywire. So, this is interesting. This is connecting the dots again. It's evidence for the functional problems that people have, just like with Mar. But, we can't just rely on these simple, observations from medicine because, this part of the brain is damaged, this function is impaired, therefore this function lives here exclusively, Because the question is, is this sinkhole swallowing a house or is it swallowing a highway, right? Is the damage destroying the entire, like, of one very specific thing, or is it screwing up an entire, like channel, an entire capacity, right? How much is damaged when this area of the brain is damaged? So the question is, how does the information actually move around in the brain on the neurons? Does it just stop here? Does it branch out again? What is the delineation for this? Okay, now warning. We're getting into animal studies now. And this kind of work involves animal subjects. And it involves what are called lesions, which are irreparable damage inflicted on brains. Not just brains, any tissue can be lesioned. But this work is not kind. It's not cruel. It's as minimally cruel as it can be to these animals, but it's not kind to them. So I've known people who have left biological psych because of this, because they've had to deal with too many rats, too many monkeys that they have to hurt in this way. And but the key here is whether or not you object to this. That's totally valid. I think there's valid reasons to say that you couldn't be a part of this yourself, that you couldn't, you don't support it continuing to happen. And there's also valid reasons to say, hey, that this is actually worth the cost, right? And that's an important discussion to have in your life with people that you care about. But what we're looking at right now is a historical record. And these are things that we have discovered using these techniques to say, I will take animals that have brains that work in ways that are similar enough to ours, and I will understand the way the brain works using these animals. Again, it can be a little bit squicky to deal with. There's some moral questions, but we can also learn quite a lot. And it is important to understand these things, especially because of the cost, not just because it's useful. So moving forward, we're going to talk about some monkey studies and some damage to the brains to understand how the information is processed. Brace yourself, here we go. So the technique used in these studies is about cross-lesion disconnection. And the idea is trying to get to the question of, does damage to each of these areas that we're interested in always impair functionality? And based on this idea that brains are divided up into two different hemispheres, as you may be familiar with already. And there's lots of redundancy on both sides. If I have something going on the left side, often there's a mirror going on the right. Now, you may have heard, of course, brains are... there's a division of labor, and the left side is responsible for one thing while the right side is responsible for others. I will tell you right now, downplay that a little bit. The left-brainedness and right-brainedness is just more horoscopes for people to latch on to. The fact is that your brain is always working in concert, but there is some redundancy on both sides. And the corpus callosum is what connects the two sides, right? It's the sort of the information superhighway between the sides of the brain. It connects every area on the left half to every kind of matching area on the right, and some cross connections between, right? It is a big, fibrous, internal highway for information between the two halves. So, how do we capture spatial reasoning pathways? What's the thought? So, information is flowing from the occipital lobe for the basic visual processing, to the parietal lobe for some spatial processing, and then to other areas of the brain for understanding, like reasoning about this information, deciding on what actions to take, that kind of stuff. So, here's the landmark task that the monkeys were given. The monkey will receive more food for going to the food well closest to the cylinder, right? And it's a very simple, these kinds of studies are nice because the complexity of the task doesn't need to be large for it. In fact, it's often best to control. So it's saying the landmark is very easy to spatially reason about. The question is, when does that spatial reasoning break down? And to answer this, we kind of move backwards through what we know, right? So in the landmark experiment, there are 3 stages. The very first stage is to lesion the parietal cortex on one side, right? So this is what we already know about, sort of, right? Okay, if they have brain damage in the parietal cortex, there is some issue with processing information in the world. It makes it harder, right? It's harder to, sorry, not just the information, but the spatial understanding of the world around them. We know that from the medical reports, right? But now the study works backward and it says, okay, does just that actually cause this kind of deficiency, this inability to reason spatially properly, or is it some other issue? So then they work backwards and they then lesion the visual cortex on the other side, which is what they expected, that the information is moving across these different regions. And then finally, if is that causing the issue? So if I have damage to both of those sections, is that causing the issue? We think there's one pathway here of primary visual cortex to parietal cortex on the other side. If I've hurt both of those regions, does this still happen? The final question is, if they then lesion the corpus callosum and say, I'm going to cut off the ability for these regions to talk to each other, then what happens? So the question is, when does this basic functionality break down? How much damage does there need to be for this to cease functioning? So the wild thing, the very interesting thing here, is that the monkeys actually maintained their capability of solving the landmark study, the landmark task, until the corpus callosum was cut. So the basic damage to the other areas didn't actually cease all function, right? They were able, the thought is, they were able to compensate because they still had the corpus callosum intact. So the simple damage did not impair the function until they couldn't talk across hemispheres. And this supports the idea that there's more than just a simple single location that's responsible for this. There's more than one tiny region that does all of this thing. There's some redundancy and there's some ability to function around it. Because there's more than a single train stop pathway through the brain. Now we can see this in other studies with humans nowadays. Again, we don't have to, given modern techniques, it's a little easier to access some of these questions. We don't have to cut monkey brains, right? We can do studies with other tools. So, but also it takes understanding these at a basic level to find these studies in the first place. So we'll talk about visuospatial neglect. This one's interesting to me. Patients who have damage to only one side exhibit this really strange activity. So in the first study, they're asked to mark all the lines with a cross. And the second study that we'll talk about here very briefly, they're asked to just locate all the T-shaped angles. Regardless of the task, the idea is that you present them with a field of objects and they recognize it. Oh yeah, there's a field of objects. There's a bunch of stuff there. And they can understand certain things about it. But if you ask them, okay, do this task, go until you are done. When they were asked to draw lines, bisecting the lines that they see, even though they can sort of comprehend that there is a full field of information here, when it came to them having to spatially solve how to interact with this field, they can't. They can only do one side. And even that, they kind of missed a few. They're asked, go until you're done. They do, and this is what the result is. there is a huge amount of this field of lines that they have not yet crossed. They have not yet done the task on. And that's because one side of the parietal system is not getting the info. Or rather, it's not able to pass on the info to the other systems that need it. They think they're done because there's nothing left for them to solve. Even works when you're not doing hands, even if you're just kind of parsing it. Look at each of these shapes, tell me if it's a T or not. Find all the T's, locate the T's for me. This is the eye tracking data. They don't even look at that side. They can't begin to process that kind of information about this. They just don't use it. So visuospatial neglect. If they have a particular kind of damage to the parietal lobe, they become functionally blind, even though they are not technically blind in that specific region, right? In that side of the vision. Okay, so the landmark study, it's a good one. We thank the sacrifice of these animals, and I mean that genuinely. But we found some very important things about this sort of sparks research and understanding of how does information move in the brain in a variety of ways. So, and it shows that the brain actually has a lot of redundancy. There's different regions that can compensate from one another, but also that the information does not just flow one direction or another. There's this division of labor. So, that's the landmark study. But let's take a dip into a little bit more lighthearted example. I always like this one. This is the Ebbinghaus illusion. Now, which of these feels larger? Which one appears larger to you? These orange dots. You probably know the answer is that they're both the same, right? This is an illusion. Of course they're the same. But the interesting thing is that there's been studies that show, perhaps not this exact one, but in these kinds of things, there's a strong difference in the way that you process this based on if you are asked, tell me which one is larger versus reach for the larger one. It's called the Ebbinghaus illusion. So if we're asked to form language to describe which one is larger, we make the mistake. But if we're asked to just act physically on the visual information, we don't. This is interesting. I can't promise that last screen will produce this illusion. You know, there's studies on this. Look into it if you're very curious. But the idea of these studies, the idea of these illusions, is that your different parts of your brain are doing different stuff with the same information. When you have to run it through language, it's prone to different mistakes and limitations and capabilities than it is if you're trying to run it through physical action, right? Wild stuff. Okay. So, this has been our first foray into digging into the brain, right? Examining how do cognitive functions actually live in the physical brain itself. And identifying that there's more than just localized function. You know, there's important connectivity between different regions. Okay, final thought. Think about your experience of this lecture. How many different ways was the information processed? Which lobes? What functions were you employing? All right, that's it. Take care.